So today, as Renee said, I will talk to you about some of our work. So it's sort of like three parts, a bit in general about our work, then a bit about work in lectins specifically, and that segues into our work in Glyco 3D that will motivate it, as you will see in a second. But first, some sort of general overview. As Renee already said, my group works pretty broadly in computational glycobiology, from statistics, data science, and AI, but then also we do some wet lab mainly in milk oligosaccharides and some more sort of functional analysis of glycans and their roles. But from method development to application, we have a lot of interest in really understanding what make glycans tick, basically. So what are sort of the sequence, structure, function relationships in complex carbohydrates? And we need to develop tools to better understand this, and that's what we do, and then we apply those to various biological questions. And we do that mainly with this computational hub that we call Glycowork, which is a Python package that we have developed and continue to maintain now for, I think, 4 years now, or a bit more than 4 years. So it's an open access Python package that anyone can freely download and use. And basically every project that we do in the group, if it's something useful that we think is also generally useful to the community, then we add or improve that in Glycowork. So that includes data sets that we have curated and now store there. That includes models that we have developed and store there as trained versions to predict certain things. But it also includes many workflows for working with glycomics data, for instance, or plotting, etc. So just one example here would be our data sets that we host, to be curate and host. So we have a lot of interest in associations, so glycan species associations or tissue associations, because that really helps us to benchmark machine learning models to predict that from a sequence. But as I will talk a bit more, also a lot of lectin-glycan interactions, mainly glycan array derived, but that is also something that we continuously increase. And we do that to provide that for the field, of course, but we also do it because we need it for our models. So whenever we do an AI model to predict something from glycan sequences, then we need a lot of data to train such a model. And we need data that is, if not standardized, then at least homogenized to such an extent that we can combine it all into one big data set, because each individual experiment is not enough to train a machine learning model, basically. So you need to combine lots of information, and it's a lot easier for us to do that if we have control over the data. That's why we curate it. We also produce a lot of tools that we ourselves initially need, but then we make that available, hopefully, to the community that also some other people might find them useful. So this is Glycodraw, for instance, which we use to draw SNFG conform glycan cartoons as vector graphics, which we then also use in figures, for instance, as you can see here, where we can automatically annotate figures basically with the SNFG cartoons as vector graphics. So it's very nice to make figures with that if you're particular about how something should look. And very happy to say that GlycoShape, for instance, which I will talk more about later, also uses Glycodraw under the hood to display these nice little cartoons. Hopefully more sources will use that. That will be very satisfying to see. So transitioning on to some of our work more on lectins. So the reason why we work on lectins is because we want to understand glycans, of course. So as probably many of you know, lectins are very prevalent in many different facets of biology. So here, for instance, in the immune system and in cancer, where you have interactions between lectins and complex carbohydrates in a sequence-specific manner, where you have basically the readers of the glycocode, if you wish, but you also have the exploitation of the glycocode, where you have lectins that are derived from pathogens, or at least some kind of microbial life form that use our glycans as receptors for their lectins, and use that to either adhere or invade our cells, basically. Again, all of that happens in a somewhat sequence-specific manner, depending on the lectin, how specific that is. But there is a certain interplay, there is a certain, if not rule-based, then at least association-based connection there that we maybe can understand and then eventually exploit. So we find ourselves at this problem, basically, that we have a lectin from, let's say, a virus or a microbe, and we have a bunch of glycans. So let's say this is the glycome, often a lung of a cell in the lung, for instance, and then you want to find out, does anything bind, and if yes, what is actually bound, right? So you can do that experimentally, of course. And maybe you do that and you find out that a certain number of glycans will be bound by that lectin and some other glycans will not be bound. Always with the caveat, of course, that bound and not bound are very strict categories, and sufficiently, if you add something in a sufficient concentration, everything binds. But you can still, if you make a threshold, at some point you can say something is binding and something is not binding. Now, if you are a glycobiologist and you look at these nice SNFG cartoons, you might notice some patterns. So these sequences all look very different, but they all have a fucose, and the non-binders have no fucose. So just looking at that naively, you would say this lectin binds fucose, right? And I mean, this is a very striking enrichment, so that might not be a wrong conclusion. But as we'll see later, that might not be the right conclusion either. In any case, the problem is that this is actually not the issue. The issue is that you have many proteins and you have many glycans, and you can't necessarily do this in a very nice screening manner for both domains. You can't screen 1,000 proteins against 1,000 glycans and hope to be done before you retire, at least not with technologies that we have today. That's where our prediction really comes in, because it allows us to screen these interactions in silico, hypothesize something, and then test that in a glycan array format or something like that. To get a prediction, again, we need data. In this case, because it's an interaction between glycans and proteins, we need data for that interaction. We use glycan microarrays for that predominantly. So this is this technique where you immobilize glycans and then you add soluble lectins that are either labeled themselves or are at least tagged in a way that you can, via secondary detection, label the lectin in some manner. And when you add it to the slide, then you can basically measure to which spot does the lectin bind, in which intensity or which relative amount. You can, since you know where each glycan sits on that array, you can then link that information with the glycan sequence. So you get a relative fluorescence value for each glycan sequence and use that for analysis. And people have been doing that for a long time, of course, analyzing it via various means, very cool algorithms to find out what is actually the enriched binding motif in the bound glycans versus the non-bound glycans, etc. But we have sort of aggregated that information. So back when we did this in the beginning, it was around 550,000 unique interactions, where unique means unique protein sequence, unique glycan sequence, and then one value that is aggregated across many different experiments and via mean or median aggregation, etc. Nowadays we have around 800,000 of those, so it's increasing, which is good because it's not only increasing the absolute number of interactions, but also the diversity. So we get to see more lectins, more glycan sequences, etc. In any case, back when we did this the first time, we used this information to build this model that we call LectinOracle, that takes in glycan information and protein information to predict whether these will bind, using a graph neural network for the glycans that we have developed and using an existing language model for proteins that we then combine together into one model, basically. This is great. It's now one of the standard benchmark tasks for evaluating new Glyco-AI models. So this is from a benchmark paper that a group in Canada has done about a year ago or so, where they have accumulated different tasks, where one of them is protein-glycan interaction prediction. And then whenever sort of like a new model comes out, the idea is that you run all these tasks through that model to see, is this model actually better than existing models, or is it better for some tasks but not for others, and all that. So this really benefits from all these data sets that we and others curate. One of the reasons why we do it, of course. But it's only a subset of tasks that you could do. So we're still sort of developing new tasks, and we're currently working on a new benchmark that will then evaluate some different tasks. One of which I will talk about later, this torsion angle prediction, but also some others that we have data sets for, like tissue expression and mass spec fragmentation spectrum prediction. But we'll talk about that in a future date. So just about LectinOracle before I segue into the 3D angle. when we developed this model, the idea was really to use this for unseen glycans or proteins. Basically, to find new lectins or to find new glycan receptors for existing lectins, etc. So we have, at the time, we validated this quite extensively to see that what you see here is basically a correlation of the experimentally determined binding on the y-axis and the predicted binding on the x-axis for data points that have not been seen by the model. So we usually see moderate to good correlations, which is good. And we then can apply this also to sort of metagenomics data to mine hypothesized associations of the lectins of the microbes in certain biological settings with their predicted binding motifs. So that's all nice and well. It's also nice that other people have started to use LectinOracle for exactly this purpose, to basically biomine genomes for hypothesized lectins with desired binding specificity. So in this case, the desired binding specificity was against cancer-enriched glycans that these authors here have looked for in our Glycowork datasets, and then you look for lectins that are predicted to bind those glycans as anti-tumor agents, which has worked very nicely with the cytotoxic lectin that they then have found and characterized here in this paper. So that's good. But what we found recently is really that while we work a lot with sequences are not necessarily a one-to-one correspondence to a structure motif. And that matters for lectins, because a lectin doesn't bind a cartoon, a lectin binds an actual 3D molecule. So the actual 3D coordinates are important. So what we did here, so just to walk you through that for one lectin, this is Bamble, a lectin from a nice little microbe here, which binds fucose. Not exactly all fucose linkages, but as you can see, it's a bit promiscuous in terms of what kind of fucose it binds. The fucose in our example here is always terminal, because the lectins that we look at here, the glycans that we look at here, are very mammalian dominant, basically. So we're looking at fucose motifs that are always terminal. All of the dots in these two plots have or are glycans with this motif. So we only looked at glycans that contain the binding motif. And then on the y-axis, you see the experimentally determined binding. And on the x-axis, you see the structural characteristics of this motif in that glycan. So if sequence would be sufficient to explain binding, then all of the sequences that have the sequence motif that is bound should have high binding, and there should be no correlation with the x-axis whatsoever. That's not what we find. Actually, most glycans that contain the binding motif, that is the bona fide binding motif, do not bind Bamble at all. And now you might ask yourself why that is. What we find here is that only the fucose motifs that are actually available on the surface of the glycan are being bound. Whereas if you have a fucose that's maybe a bit buried or maybe not so accessible to the lectin, that's not so much bound, which sounds kind of intuitive when you think about it, but it's something that is not so intuitive when you look at the cartoon, because the cartoon doesn't necessarily tell you whether the fucose is buried or not, but you need it to be accessible in order to be bound. So this is it came to us as a revelation. Other people have of course know this for a very long time, but sequence is not equal to structure. So that's really when we look at these great databases like GlycoShape from Melissa Fada's group, where they collect molecular dynamics simulated glycan structures. You can see here, if you look at the different conformationally sampled clusters that they nicely align here, you see that there is quite some difference in the positions of these monosaccharides in the different conformational clusters. So a lot of flexibility, which is usually not considered if you do analyses on the sequence level, which we and many others do. But it might explain lectin binding, which is a great opportunity for us, of course. So we have sort of this disconnect where, again, it's not that everyone has this disconnect, but at least we and many other glycoinformaticians have that disconnect, I think, that we tend to work a lot with sequences because sequences are available, sequences are nice and tractable. They are they have a long history, of course, in bioinformatics in general, but then you have coordinates on the other sort of side of the divide, which are more used for structures, which again also have a very long history. But it was an opportunity for us to explore new frontiers as well, which we then combined into this new package that we developed that's called GlyContact, and that's meant to analyze structures from the perspective of someone who likes to work with sequences. So you basically interact with sequences, but you actually analyze structures, which is what I like to do. So the idea is that the input here are PDB files pretty much. We started out with PDB files from GlycoShape, so to use these molecular dynamic simulated glycan structures, but the code itself works also for unilectin 3D structures or PDB or general PDB structures. As long as there is a glycan in your PDB file, the code will extract the glycan coordinates and then transform them into a format that we can work with to do all kinds of stuff, like here are nice little contact maps, which of course are more possible with the GlycoShape dynamic information, where you can basically monitor which contacts between two monosaccharides change the most between conformational sampling. So to have a sort of a sense of where in the molecule does the change happen, the flexibility, which is useful to sort of gain this intuition. But you can also enrich existing cartoon depictions of glycans by things like surface availability. So here we have a classic Glycodraw drawing of this nice N-glycan, but then you have this color gradient to indicate which monosaccharide here is surface available to which extent. So maybe not so surprising, the salic acids here at the terminus are very surface available, whereas the inner mannoses maybe are less so. So it's a way to visualize, it's a way to convey certain structural parameters of these sequences, and it might teach you, a surprise or two if you look at some of these glycans. It's also a great opportunity for us to do some 3D visualizations. Again, this is since GlyContact uses Glycowork behind the scenes, it's sort of like a downstream package of Glycowork. We can make use of a lot of the functionality that we've built over the years. So for instance, you can use GlyContact with any nomenclature that you choose, and then it will, under the hood, convert it into something that we can work with. So here, for instance, just using it with a work sequence where I just, in my careless sloppiness, just added lots of spaces, and it's fine with that. Or using here common names, and then you get this nice 3D rotatable structure that is just representing a conformational snapshot, of course, here in this particular visualization. But it still allows you to sort of rotate it around and again gain some intuition about the whole thing. Since we have all this nice functionality on the sequence level, we can now very easily use that to probe structures for properties. So for instance, in Glycowork, we use basically molecular graphs to work with glycans. So we have very easy and powerful access to what is terminal, what is internal, and all kinds of other information in the sequence, which maybe, in just a pure text format is hard to get at scale. But we can then ask very simple questions like taking the monosaccharide galactose and taking all the glycan structures that we have, we can compare the surface availability. And I should probably mention SASA is just the solvent accessible surface area. So just think of it as the surface area that matters pretty much. And then you get results that again sort of are aligned with intuition, but are good sanity checks, like that terminal galactose monosaccharides are on average more surface available than internal galactose residues, which is good to see that we capture these trends that we expect, basically. Because then we can go on and do more ambitious analyses. So we do something that's called twin analysis, because one thing that bugged me for a long time in analyzing glycans is that you have so many confounders. So whenever we compare two groups of glycans, they differ in many different ways. So I don't know, let's say you want to do like motif enrichments or something like that. You always have so many correlates of other things in the sequence that you don't care about, but that they also differ between your groups. So that's why we here engaged in paired analysis, basically. So we looked at sequences where we had structures for those sequences, and we wanted to find pairs of sequences that only differ in one specific characteristic. Here, for example, in core-fucosylation, so where we have sequences that are not core-fucosylated, and then the same sequence but core-fucosylated, and then do that for here, I think it's close to 50 sequence pairs, basically, where we have that. So in this paired analysis, you control for all confounders. Classic statistics, nothing revolutionary, but very powerful in the case of glycans, I think. Because what it does allow us now is to look at the basically only the effect of the core fucose without all the confounders. What we look at here is basically the overall surface area of the glycan, but not looking at the reducing end, because that will be cheating, because, you know, of course you expand the surface area by adding the fucose, so you have to take that away. But you can see that if you do that, then on average, the core fucose addition slightly decreases the average surface availability here. And you can, since it's so powerful, you can do that even with very few data points. And as long as it's very consistent, you still get significant results, even, you know, if you just have like a handful of pairs, basically. But this here is the impact on xylose addition in what you have in plant glycans, for instance, where it opens up the end glycans a bit. And you can see that here in the surface addition, surface area analysis. What we also can do is, and we really like to do that as well, is to look at sequence motifs and see how they behave in different sequence contexts. So here is sialolacnac, which we basically looked at in different glycan classes, because glycan classes present very different sequence contexts, present very different structural contexts. That's why lectins sometimes have glycan class specificity, because it's a different 3D context. And you can see here that the sialolacnac motif actually behaves differently, for instance, in N and O glycans, because again, it is present in a different sequence and structure context, where in N glycans, you on average see it being less surface available than in O glycans, as you can see here. We see typically the same effects for the GC and the AC variants of motifs. At least we haven't encountered discrepancies here so far. So we come back to this idea of you know, looking at lectins from a glyco-3D perspective, where you now hopefully can appreciate this idea that increased surface availability also increases the likelihood of a binding event between the lectin and the glycan. But this is just Bamble, of course. We looked at many more lectins, and if you look at the x-axis, this is the correlation of the surface area and the binding. And you can see that most lectins have a positive correlation for that. So most lectins prefer a surface available version of their binding motif. So here, Bamble, just for orienting yourself. There's also some relationship here with flexibility that is a bit harder to disentangle, but the surface area is something that is, I think, very intuitive to understand. So it's important for you to know, basically, for a given glycan, whether this is available or not. So if it's important for you to know, we thought it's probably also important for the model to know, because if you give our poor LectinOracle model just sequences and expect it to, you know, just work with that. So we thought, let's make the life of LectinOracle a bit easier, and also give it some of the structural parameters that we can calculate from the PDB files and infuse into the graphs that we then hand over to LectinOracle in the graph neural network. So basically, the monosaccharides gain structural attributes that we calculate. The linkages gain these torsion angles. So basically a measure of how they are oriented, how the two monosaccharides are oriented toward each other. And then that is available to the model for prediction. And if we compare that to the base LectinOracle, then you can see that for many lectins, we get an improvement in the correlation between experiment and prediction. Some lectins go the other way. Those are usually the lectins that bind, in our experience, rare conformational states. So they are giving that information is actually skewing the model into a wrong direction, because you give it the information where the more common conformational states are. So you're guiding it away from what is actually being bound. In any case, it's a net increase for the performance, so that's really good. So we are very excited about that. But if I'm being honest with you, I would have expected an even bigger increase because it's structural information. It should, you know, it should blow performance through the roof. So we went back sort of to the drawing board, and we thought that probably some of that information is already contained in the sequence, and the model has access to it, and has learned some of these associations. It's just make it easier to provide it explicitly rather than implicitly in the sequence. But then, if you make this argument really, you bring it to the end, you can then say, if the structure is contained in the sequence, does that mean that we can predict structure characteristics from sequence? And that's sort of what is the last piece here that I want to talk about. Where, of course, the main challenge is that structure is really a problematic word for glycans, because structure implies sort of a static kind of 3D orientation, which is very much not true for glycans. Very flexible, a lot of different conformational areas that they can sample, right? So there is not one answer to a sequence. So you have this classic one-to-many mapping, which machine learning models hate, where you have one sequence, you have multiple answers, and you don't even have the same number of answers, because if we want to use this word of conformer as sort of like a conformational cluster representative, then depending on the sequence, there might be more or fewer such conformers. So in the end, if you would train such a model, what it would learn to predict is to learn some kind of average, which is true for none of the conformational states. So you don't want to do that. That would not be physiological, so it would not be a very useful model. So what you need to do, as usual in AI, is you basically need to rephrase it into something that is one-to-one, because one-to-one is a very nice problem. You want for one sequence, you want one answer, because that you can train well. So to do that, what you need to sort of put in between the sequence and the conformer is the conformational ensemble. So basically the entire space that the glycan can sample for a specific sequence, because then you have a one-to-one mapping. So the conformational ensemble gives rise to all the conformers, and then you predict the conformational ensemble, which is sort of the machine learning person way of saying, job done, next problem. Until you realize that conformational ensemble is not really a box that you can predict very nicely. So you need to first think about what is a conformational ensemble. And in a way, an ensemble is nothing more or less than a distribution. In our case, since we're interested in torsion angles, it's a distribution of torsion angles. And if you have multiple conformational states, then it's a multimodal distribution, because you can have multiple peaks, basically. Now, if it's a distribution, and here it would be a multimodal distribution, just for your reference, if it's a distribution, we don't need to predict the distribution per se. We predict the parameters that describe or generate the distribution. So if we look here at a standard normal distribution, we would predict maybe like the mean value or standard deviation, and then you can reconstruct that those distributions from those parameters. This would be a classic Gaussian mixture model. It's very established for various purposes. But basically, that's what we want. We want to have a distribution which allows multiple states to coexist, which would then be our ensemble. That's almost perfect. The only annoying thing now is that torsion angles are angles, and angles are not normally distributed. Because if you have an angle of 359 degrees and an angle of 1 degree, that's almost the same. That's not what the normal distribution wants. So what you in the end want is a von Mises distribution, which is basically the Gaussian distribution for angles. So it's arranged around a circle, as you can see here, and now we have this nice property that 359 degrees and 1 degree are almost the same. But apart from that, it's very similar. So it's really the most simple circular distribution that you can imagine. And then what you have is a von Mises mixture model, where we now, for each linkage, we predict the parameters that we can plug into this nice little equation here to actually get out the distribution, which is nothing more than a probability density of the torsion angles that this disaccharide can have, basically. And then which exact torsion angle is realized does not only depend on the sequence context, but also the conformational cluster in which you currently find yourself in. But it's something that works extremely well. So when we look here, these are the prediction errors, which I should mention, this is our standard graph neural network. So the architecture of the model is very similar to what we used to do, just the output is something very different. The output is now tightly linked to quite a complicated statistical procedure, which I think is a nice fusion in a sense. And then what you predict are these parameters that you can use to generate phi and psi and all these other angles. And as you can see here with the prediction error, this von Mises SweetNet Frankensteinian monster basically creates very nice predictions for these torsion angles, much better than directly predicting the torsion angles, which, as you can see, barely outperforms mean or median baselines. So you really need to consider these multi-conformational state problem that I just talked about. So if we now actually look at what that looks like from a Ramachandran plot, where you can plot basically the allowed or permitted torsion angles or conformational states of a disaccharide here with chitobios. These are the experimental torsion angles basically for chitobios, and then these are the predicted angles. And the only reason why that looks different is because we sampled 5 times as many data points here for the predicted one than for the experimental one. But it's really a great match. I should also mention that we can do very similar things for predicting the surface area. Here you see a correlation of the true surface area of each monosaccharide, and on the y-axis then the predicted surface area of or solvent accessible surface area of each monosaccharide, and it's a great correlation. So it's really something that is very contained in the sequence, where we now of course this is now sort of like a weighted surface area across conformers. I should also mention that this model is capable of doing zero-shot prediction, where we can give it disaccharides that the model has never seen, and we get so this would be here on that column, that those are the experimental torsion angles, and then here are the predicted torsion angles, and you can see very nicely, like here for this glucose disaccharide, where we very nicely capture the cluster of torsion angle combinations that are permissible in this context. So very nice. So for disaccharide prediction, this is pretty much, I think, what you would need in a sense. So we are currently very interested in exploring this further and seeing what else except for torsion angles can you do with such a nice combination of statistics and AI. So with this, I come to my conclusion. The conclusion is AI and data science are great for glycobiology and for glycoinformatics in particular. Still, there is a lot to be done, of course, especially because everything glycan-related requires dedicated analysis, requires custom tools and custom platforms, because we are special little snowflakes, unfortunately. But that also is fun, so there's still a lot to do here, and now also joined by our 3D arm and angle as well. With that, I want to, of course, thank my group, our collaborators, our funding agencies, and you for your attention.